Welcome to Biology, the science study of life. Generation of lymphocyte diversity by gene rearrangement. Recall that B cells and T cells recognize specific epitopes on antigens via their antigen receptors. A single B cell or T cell bear about 100,000 of these receptors or identical. However, if we were to compare one B cell to another or one T cell to another, the chances that both would have the same B cell receptor or T cell receptor are very slim. The variable regions at the tip of each antigen receptor chain, which form the antigen binding site, account for the diversity of lymphocytes. The sequence of amino acids in these regions varies from cell to cell and determines the specificity of an antigen receptor. The variability of these regions and therefore the possible antigen specificity is enormous. It is estimated that each person has as many as 1 million different B cells and 10 million different T cells, each with a particular antigen binding specificity. Thus, our repertoire of lymphocytes can respond to an enormous number of different antigens. At the core of lymphocyte diversity are the unique genes that encode the antigen receptor chains. These genes consist of numerous coding gene segments that undergo random permanent rearrangement, forming functional genes that can be expressed as receptor chains. The light and heavy chains of the B cell receptor and of secreted antibody are encoded by the same genes called immunoglobulins or called immunoglobulin Ig genes. The immunoglobulin light chain gene contains a series of 40 variable V gene segments separated by a long stretch of DNA from 5 joining G gene segments. Beyond the G gene segments is an intron. Notice that intron. Notice this figure, followed by a single exon, designated C because it codes for the constant region of the light chain. In this state, the light chain gene is not functional. However, early in B cell development, a set of enzymes, collectively called recombinase, links one V gene segments to one G gene segments, eliminating the long stretch of DNA between them and forming a single exon that is part of V and part G. Recombinase acts randomly, that is, it can link any one of the 40 V gene segments to any one of the 5 G genes or gene segments. So for the light chain gene, there are 20 or 2,000 possible 200 possible gene products, 40 V by 5 G, which means 200. In any one cell, however, only one of the 200 possible light chains is made. Once V and G rearrangement has occurred, the gene can be transcribed. The resulting pre-mRNA is processed, forming mRNA that is translated into a light chain containing a variable region and a constant region. The light chains produced in this way combine randomly with the heavy chains that are similarly produced, forming the B cells antigen receptors. Notice as shown in this video. Closer look. Testing and removal of self-reactive lymphocytes. Because the rearrangements of antigen receptor genes are random, a developing lymphocyte may end up with antigen receptors that are specific for some of the body's own molecules. As B cells and T cells are maturing in the bone marrow and thymus, respectively, their antigen receptors are tested for potential self-reactivity. For example, the receptors of maturing T cells are tested against class 1 and second class MHC molecules, which are both expressed at light levels in the thymus. For the most part, lymphocytes bearing receptors specific for molecules already present in the body are either destroyed by apoptosis or rendered non-functional, leaving only lymphocytes that react to foreign non-self molecules. That capacity to distinguish self from non-self continues to develop even as the cells migrate to lymphoid organs. Thus, the body normally has no major lymphocytes that react against self components. The immune system exhibits the critical feature of self-tolerance. Failure of self-tolerance can lead to autoimmune diseases such as multiple sclerosis, as shown or notice also this figure, colonial selection of B cells. Colonial selection of lymphocytes, a soluble antigen or an antigen present on the surface of a microbe-infected body cell or cancer cell, encounters a large repertoire of B cells and T cells in the body. However, a given antigen interacts only with the relatively few lymphocytes bearing receptors specific for epitopes on the antigen. The selection of B cell or T cell by an antigen activates the lymphocyte, stimulating it to divide many times and to differentiate, forming two colonies of daughter cells. One notice that closer look, one colon consists of a large number of short-lived effector cells that combat the same antigen. The nature and function of the effector cells depend on whether the lymphocyte selected is helper T cell or a T cell, a cytotoxic T cell, or a B cell. The other colon consists of memory cells, long-lived cells bearing receptors specific for the same inducing antigen. This antigen-driven cloning of lymphocytes is called colonial selection. The concept of colonial selection is so fundamental to understanding acquired immunity that is worth restating each antigen by binding to specific receptors. Selectively activates a tiny fraction of cells from the body's diverse pool of lymphocytes. This relatively small number of selected cells given rise to colonies of thousands of cells, all specific for and dedicated to eliminating that antigen. The selective proliferation and differentiation of lymphocyte that occur the first time the body is exposed to a particular antigen represent the primary immune response. The primary response does not peak until about 10 to 17 days after the initial exposure to the antigen. During this time, selected B cells are generated antibody selected or secreting effector B cells called plasma cells, and selected T cells are activated to their effector forms, which have distinct functions. While these effector cells are developing, a stricken individual may become ill. Eventually, symptoms of illness diminish and disappear as antibodies and effector T cells clear the antigen from the body. If an individual is exposed again the same antigen, the response is faster, typically only 2 to 7 days, of a greater magnitude and more prolonged. This is the secondary immune response. Measures of antibody concentrations in the blood serum over time show clearly the difference between the primary and secondary immune responses. As shown this figure, notice that this diagram, in addition to being more numerous antibodies produced in the secondary immune response, tend to have greater affinity to that for the antigen than those secreted in the primary immune response. The immune systems or the immune system's capacity to generate secondary immune response, called immunological memory, depends on the colonies of long-lived T and B memory cells generated following initial exposure to an antigen. These memory cells are poised to proliferate and differentiate rapidly when they later contact the same antigen. The long-term protection developed after exposure to a pathogen was recognized 2,400 years ago by Thucydides or Thucydides of Athens or of Athens, who described how these, although sick and dying of plague, were carried for by others who had recovered, for no one was ever attacked a second time. Back to a culinary selection of B cells. Notice this figure, B, A, and B cells is selected by an antigen to proliferate and differentiate into memory B cells and antibody secreting plasma cell. All the P cells whose receptors had a different specificity indicated by different shapes and colors do not respond to this antigen. Lymphocytes undergo a similar process generating memory T cells and effector T cells. Notice these are B cells that differ in antigen specificity. Notice that. And this antigen molecules bind to the antigen receptors of only one of the three B cells shown. Notice also this step that selected B cell proliferates, forming a colon of identical cells bearing receptors for the selecting antigen. Notice here this step also colon of plasma cells. Some proliferating cells develop into short-lived plasma cells that secrete antibodies specific for the antigen. Notice these are antibody molecules. Notice that. And colon of memory cells here, some proliferating cells develop into long-lived memory cells that can respond rapidly upon subsequent exposure to the same antigen. Okay, thanks.